audible to you guys?
Yeah, I'm audible.
Am I clear?
Yeah, I'm clear.
Should we wait or should we begin the class?
Ma'am, just 2 more minutes, ma'am. We'll start the class.
Okay.
As I posted the link late and people might be delayed joining. We'll join.
Okay.
Yeah, ma'am, we can start the class.
Okay. So we had discussed about the basic in-field protocols that need to be followed at the crime scene while dealing with biological and DNA examination. Right now, I'll be discussing very briefly the procedures that we are undertaking in the laboratory for processing the samples that we have collected at the scene of crime. So what happens after you have sent the samples to FSL? What is happening at FSL? Why are the sample collection so important, and why should we maintain all that contamination control and everything? You'll get an idea over here. So basically what we do is we extract the DNA. We quantify the amount of DNA that is present. The quantity of the DNA needs to be established, right? You cannot just extract and proceed without understanding whether there is any DNA in it or not. So you need to be very sure that there is DNA and what is the quantity of DNA that is present. Amplification of DNA, we need to increase the quantity of DNA for the detectable quantity. So every instrument has a detectable quantity below which it cannot give signal, or it cannot give you the result of the amount of markers that need to be amplified. So for that reason, we amplify the DNA that we receive at the scene of crime samples so that we get the appropriate DNA profile results further down the process. Then we separate the DNA double strands. As we all know, DNA is a double helical structure. It is something like this. We have a double helix here, bounded by bonds. So we break these bonds and we we separate the DNA into single stranded structures. Now, we analyze these byproducts and we interpret the results. We identify the presence of the markers, the location of the markers. Basically, as per CODIS right now, you must be knowing what CODIS is. Combined DNA Identification System. CODIS. Are you able to see what I'm writing on the screen? Are you able to see what I'm writing on the screen?
Yes, ma'am.
Okay. So we use CODIS system. It is combined DNA identification system. Right now, CODIS is working on 26 plus 1 markers. 26 markers are different, and this one that we are saying is amylogenin. Amylogenin. This is basically a gender discriminatory marker. So CODIS system is currently working on 26 plus markers in all the FSN, right? So what is the location of this marker out of these 26 markers? What is the length of the marker? What is the exact allele and the location where these markers are present in that particular individual? Those are interpreted after separation and extraction, and ultimately the DNA quality is checked. The report is reviewed, and we estimate whether the quality of the DNA that has been obtained is a good quality to report or not. Clear?
Yes.
So, what are the general principles of extraction? The first step that we undertake while extracting DNA is breaking open the cell to release the DNA. So, any cell of any body is containing cell wall, cytoplasm, and a lot of other aspects that is there, and embedded inside is the nucleus, which again is covered by a membrane inside which the DNA is present. So in order to analyze or in order to interpret DNA, we have to break open all these walls and we have to release the DNA from inside the nucleus. So the first step of DNA extraction is the breaking open of the cells so that the nucleus is exposed. So what is the component that we are using for separation of extraction of DNA? You need to keep a notebook with you and write down whatever I'm telling you, because it's not there in the PPT. Just take notes, the important words that I'm using. What we use is SDS, something known as SDS. What is SDS? SDS is sodium dodecyl sulfate. Sodium dodecyl sulfate is a detergent, just like the detergent that we are using to wash clothes or soaps that we are using to take bath. Sodium dodecyl sulfate is also a detergent that breaks down the lipid and the cell membrane of the nuclei and releases the DNA. Clear? Is it clear?
Yes, ma'am.
That was the um, separation
What?
process, right?
What?
SDS page was the separation process.
SDS page is a separate process. SDS page is usually used for proteins. SDS is a chemical, and that chemical is used as a detergent. There is a difference between sodium dodecyl sulfate and SDS page method or instrumentation. SDS page has nothing to do with DNA right now. I'm just talking about the chemical here. The detergent is something that is causing the lysis of the cell. This breaking down of the cell is also known as the lysis of the cell. And this lysis is basically caused by a detergent. And the name of the detergent is SDS. Full form is sodium dodecyl sulfate. It is a chemical, not page. SDS page is a instrumental, or you can say it is a technique used in case of protein analysis, not in case of DNA extraction. Am I clear?
Yes, ma'am.
Is it clear?
Yes,
Yes,
ma'am.
ma'am.
Good. Don't confuse things. You should understand what we are talking about. SDS is a chemical, SDS page is a technique. Just like ELISA is a technique, or PCR is a technique, SDS page is just a technique. It is not used in case of DNA extraction. Okay. Now the next step is separation. Separation of the DNA from all the protein and other cellular debris. So once the cell is being broken, there are a lot of things that is coming along with it. The cell debris, or all the cytoplasm components, or the mitochondria, or the centrioles. Everything that is there inside the cell, it is going to come off, right? So how do we separate this debris from the DNA that is actually needed? We use for the protein, we use a protein digesting enzyme. We use a protein digesting enzyme known as the proteinase K. This is known as proteinase K. Proteinase K is something that is always to be freshly prepared. You cannot prepare it and keep it in the fridge and then use it whenever you want to extract the DNA. It should always be freshly prepared immediately before the extraction procedure needs to be undertaken. Clear?
Yes.
You have to write these things down. These are the important things, and these are the aspects that you are going to come across when you are appearing for any competitive exam or for any interview, right? So this proteinase K is basically, it is doing what? It is degrading the DNA associated proteins and other cellular proteins. It is basically degrading it and separating it from the required DNA. Next is the precipitation of the DNA with alcohol. What do we do? To remove all the aquatic impurities, we need to isolate and separate this DNA into some alcoholic surface or substance so that only pure DNA is extracted. So whenever the DNA is separated from the cellular debris, ice cold alcohol, which is either ethanol or isopropanol, is carefully added to a DNA sample. DNA is soluble in water but insoluble in the presence of salt and alcohol. So by gently stirring the alcohol layer with a sterile pipette, a precipitate becomes visible and can be spooled out. Always remember, it is soluble in water, but it is not soluble in water in presence of alcohol and salt. So in order to achieve, or in order to obtain a very pure DNA, we use ethanol or isopropanol for the purpose of DNA precipitation. Next is cleaning. We need to clean the DNA completely so that no error is obtained during the PCR process. It is purified again using a very slightly alkaline buffer. We use a buffer and we centrifuge it, or we use vortex shaker to slightly mix the DNA in this buffer so that all the impurities, remaining impurities that are there in the DNA, they are all removed. Now, once the DNA is cleaned and separated, it is very important to confirm whether the amount of DNA, or whether whatever you have collected contains any amount of DNA or not. And if it contains any DNA, whether this DNA is suitable for creating a genetic profile, or whether it is fragmented, broken DNA, and the markers can either be identified or not. Right? So for further lab work, it is important to know the concentration and quality of DNA. Now, optical density readings are taken by a spectrophotometer that can be used to determine the concentration and purity of DNA in a sample. I hope you have heard of an instrument called nanometer. Nanometer gives you the quantity of DNA per microliter. So suppose 1 drop of 1 microliter of this purified DNA is added into the well of the nanodrop and light is made incident on it. The spectrophotometer will analyze, and based on the optical density, it is going to tell us whether 625 picogram of DNA is present in 1 microliter of that sample. Clear? Is it clear?
Okay, ma'am.
Yes.
If not this, a gel electrophoresis can be used to show the presence of DNA in your sample and give an indication to the quality. I hope you know what a gel electrophoresis is. We have a gel plate and we apply DNA in the wells, and we have it radio labeled, and when electrodes are added, the DNAs are run all across the gel, and we get profiles like this, lines like this, based on the fragments of the markers that are present. Have you done this practical in your college?
Yes, ma'am.
You're aware of what a gel electrophoresis looks like, right?
Yes, ma'am. AH, pH, capillary, those things, right?
Okay, great. So this is a centrifuge. This is how a high-speed centrifuge works. This is used for the purpose of centrifuging all the DNA. Right? So what is CODIS, as I told you? So the CODIS refers to Combined DNA Index System, which is a database in the United States consisting of profiles derived from the so-called CODIS markers. Because of their autosomal nature, CODIS markers can be used to assess whether two people are likely to be very closely related to each other or not. In particular, of these 26 alleles that comprise a person's CODIS, 13 would have been inherited from each parent. So if there are 26 markers, if a person is having 26 markers, or if a person is having 26 alleles, 13 of this is coming from the mother, 13 of this is coming from the father. Because 26 is equal to 2n and n is equal to 13, and half 13 would be from the mother, half 13 would be from the father. So this is how you can also identify paternity tests. Because on the Y chromosome or on the other autosomal chromosomes, whatever markers that has been taken up from the father, they're going to match the DNA profile. Father's marker position, child's marker position. They are going to overlap or match at a particular location and size. This is how you identify whether a person belongs to a particular relative or not. Right? So the CODIS DNA tool can be used to take a person's CODIS marker and determine some sense of person's ethnic background also. To do this, they generally perform a comparison of CODIS markers allocated from various sample populations. Now, the CODIS has two indexes. Every software, every database has index. Every database and every software, they have to work on a certain index, otherwise they will not be of any use. Every comparative software, if you are comparing a software or if you are searching a particular data in a software, it is bound to have an index. So the CODIS has two basic index: the convicted offender index, that contains the profiles of the convicted offenders or the people who have been arrested and have been convicted and they are in the jail. So they have their DNA profiles, and the forensic index, which contains DNA profiles that are received from the scene of crime. So with the help of these two indexes, the CODIS has created a data bank and has identified 26 main markers that need to be analyzed at a highly polymorphic. Always remember, these 26 markers are highly polymorphic. Polymorphic means that it is highly diversified. If there are 10 billion people all around the earth, the combination of these markers are going to be unique in each and every individual. So no two individual is going to have the same combination of these markers. So when there is a difference between each and every individual of any marker or anything that is there, it is known as a very highly polymorphic component. So these 26 markers that the CODIS is studying, or the CODIS has taken into consideration, these are very polymorphic, highly polymorphic, and so they need to be considered for determining the identity of any individual. Right? Am I clear?
Yes, ma'am.
Yes, ma'am.
So disaster management is there. So we were talking about DNA polymorphism. What? Are you trying to say something, dear? Okay. What is DNA polymorphism? Now, genetic polymorphism are the simultaneous occurrence of two or more discontinuous genotypes or alleles in a population, also known as genetic polymorphism. So basically, when two or more forms of a particular aspect or a particular species or a particular biological, what do you call it? Biological appearance can be present. So we are polymorphic. Humans are polymorphic. There are two varieties of humans, male and female. So male and female is a dimorphic existence of human. So polymorphic is if you have more varieties of humans. There definitely are. We have the Klinefelter syndrome, right? We have XXY, XYY, extra X chromosome, extra Y chromosome. So these are also the varieties. So we call that a polymorphic way of existence, right? So genetic polymorphism can determine the diversity of an individual. How many types of genetic polymorphisms are there? We have the phenotypic polymorphism, or the polymorphism of the external features like male and female. Chromosomal polymorphism is there. The shape, the size, the number of the chromosome can be different in different individuals. Protein polymorphism is also there. The protein, translated proteins during the DNA, replication, transcription, translation, that can be polymorphic, because of which, for example, somebody can eat sugar without worrying about diabetes, and somebody who is not into sugar has a constant fear of diabetes. So this protein translating polymorphism is also there. And last but not the least that we are studying in forensic is the DNA polymorphism. So there are four aspects of genetic polymorphism that exist. Now, DNA polymorphisms are the different DNA sequences among individuals or populations that exist in wide range of variations. When even a single base pair change of a particular gene can cause hundreds of its form to exist in nature. So if we have, suppose we have a gene that is TAATCGGC and then we have TCATTCGGCTACG, and then we can have TGGTCGG. So even one single nucleotide change or a single base pair change can result in the existence of multiple forms of these genes. So if a gene is having, suppose, say 10 base pairs, the number of forms in which it can be present is exponential, 10 to the power of 10. Right? So that is how polymorphism works. And there are so many genes in our body, millions of genes. Although forensic is just working on 26 markers on the genes, but the Human Genome Project has basically decoded all the genes that are present in the body. And with so many genes and so many varieties of the genes present, imagine how polymorphic an individual is all around the world. I mean, you will rarely find two individuals exactly the same, having the same DNA makeup, and not exhibit any polymorphism. I hope I'm clear with the polymorphism concept.
Yes, ma'am.
So why are the polymorphism happening? Either the length of the gene is changing, variations in the fragment length of the produced after DNA, variations in the size of a DNA fragment after PCR amplifications, and variations in the DNA sequence itself. Now we have DNA length polymorphism. DNA length polymorphism is studied under three aspects in forensic. Okay, we are studying short tandem repeats or STRs. Now STR is the advanced technique. STR is autosomal. Autosomal means it is non-sexual. It has got nothing to do with the sex chromosome. It is something that is related to the 22 chromosomes which are autosomal. So we have autosomal STRs which are diploid in nature. These autosomal STRs are diploid in nature. Why? Because these are studied as a combination of mother plus father. Mother plus father. So this is a definite indication of the presence of a particular person at a scene of crime. In addition to this, we have something known as the XSTR, which is the X chromosome, and the YSTR, which is the present on the Y chromosome. So Y chromosome is a haploid examination procedure. This can help you identify the presence of a male DNA at a particular place. But this male DNA can belong to a particular person X, or X's brother, or X's father, or X's uncle, or X's grandfather. So this is the same for all the male members of that particular family. So you cannot be 100% sure whether X is the culprit or whether X is the accused. You cannot give an absolute report on the basis of YSTR or XSTR. In case of XSTR, X is mother, X is sister. Everybody is going to have the same STR profile. It is an haploid aspect. It is not a diploid aspect. Only autosomal DNA analysis is the diploid aspect, which is allowing us to give 100% result about determining the identity of the accused. Am I clear?
Yes, ma'am.
Am I clear?
Yes, ma'am.
Okay. So next is the variable number tandem repeats or VNTRs. And then we have restriction fragment length polymorphism. So these two aspects, these two are now obsolete. We don't use it in forensic. Now STR analysis is what we are carrying out in DNA examination. And we have DNA sequence polymorphism and single nucleotide polymorphism. Now single nucleotide polymorphism again advanced, and DNA sequence polymorphism is basically indicating towards a complete gene. Now, single SNPs are also used in the case of NGS. I hope you all must have heard of the word NGS, which is also known as the next generation sequencing. Next generation sequencing is basically aiming at identifying the nucleotide by nucleotide identification. So here we are just working on 26 markers, but single, next generation sequencing will help us identify exact nucleotide to nucleotide identity or profiling of a particular individual. So if there are millions of nucleotides or millions of base pairs that is there, base pair to base pair profile we can create, and we can absolutely point out at the person who was present at the scene of crime. This is only based on the current CODIS system is only based on the 26 plus 1 markers. Say suppose we get a degraded DNA sample in which these markers are broken from the center, then we would not be able to give any result. But if we have base pair by base pair profile of the entire genome of that particular individual, even if the DNA is broken, it is easy to identify whether the person was present at the scene of crime or not. Clear? Am I clear?
Yes.
Too much of data on this page. Am I clear?
Yes, ma'am.
If you guys have any doubt, kindly stop me at that point and ask, because later then the flow is gone and then I won't be able to, I'll have to recollect what I was trying to tell. Okay. Now what is an SNP? Single nucleotide polymorphisms are basically a type of polymorphism involving variation of a single base pair. As you can see in the image here, this sequence, all the remaining sequences that are there, they're the same. Look at the other sequences that is there. They're all the same. The only thing that is changing is this nucleotide at this position. And the change of this single nucleotide is resulting in the formation of 3 different individuals here. Right? And in Human Genome Project, the scientists are studying these SNPs to correlate with disease, drug response, and other phenotypes, the existence and occurrence of different unique phenotypes, and how they are formed and the reason by which they are formed. They are studying this under the category of single nucleotide polymorphism. Right? You can see what an STR is. A short tandem repeat is a microsatellite. Remember what it is. It is a microsatellite with a repeat units of 2 to 7 base pairs. Write it down in your copy. SNP is single nucleotide. One nucleotide at one particular position. Short tandem repeats are a microsatellite consisting of 2 to 7 base pairs in length, with the number of repeats varying among individuals, making STRs effective for human identification purposes. And this is the reason why we are using 26+1 CODIS system, because this is ideal for human identification system. You can see here in the image, we have AG80, 7 repeats of AG80 and 6 repeats of AG80. In this individual, there are 4 repeats of AG80 and 19 repeats of AG80. To see the difference between the appearance of these two individuals. And in this person, we have 12 repeats of AG80 and we have 14 repeats of AG80. Suppose this AG80 is X marker. This is a marker. Marker X having 7 repeats of AG80 at site 1 and 6 repeats of AG80 at site 2. So we are getting a blood sample at the scene of crime which is having a profile like this. And we are getting 3 suspects. Once we create the profile, we'll be easily able to match the crime scene DNA profile with the individual's profile and say that, okay, this biological sample belongs to this individual and he is the culprit or he is the suspected accused. Am I clear? Is it clear?
Yes, ma'am.
Okay. Great. VNTRs. We have variable number tandem repeats. So within a gene, a short sequence of DNA repeated in tandem that vary greatly in number among the individuals, also known as the microsatellites or hypervariable region. Write this down in your copy that the short sequences of repeating DNA in tandem means in a particular manner, in a particular rhythm, just like the beat of a music. If you are having a beat of the music, the drum is playing in a particular pattern, in the same pattern of 2 repeats or 3 repeats after a particular length. In tandem, there are small microsatellites which are known as the hypervariable region that keep repeating in a particular gene. And because of it, it produces a genetic mark that is specific to each individual. And this is used in CODIS for DNA fingerprinting. I hope you're understanding what variable in a tandem means. It is repeating in a particular way, in a particular rhythm. There is a rhythmic repetition of these microsatellites in the particular marker of a particular gene on a particular chromosome of an individual. And this tandem is studied in CODIS for forensic DNA fingerprinting. Clear? Am I clear?
Yes, ma'am.
Yes, ma'am.
Take a screenshot or a photo of this page. This thing comes in your competitive exams. Difference between VNTR and STR. You will be encountering this for any MSc entrance exam or any UGC NET exam or any other competitive exam that you will undertake in case of DNA at least. It's important. You take a screenshot or a photo of this page, right? Can I change the slide? I hope you guys have taken the photograph. Should I change the page?
Won't you share the PPT?
This PPT has a lot of other information which is not needed. So, and anyways, this is a very basic PPT. This is not something that you would need. You would rather need what I'm making you write in your notebook.
Okay, ma'am.
The other two PPTs, I guess you have received. Have you guys received the other PPTs that I shared?
Only one I have received, ma'am.
Okay. Great. This is a PhD level PPT, so I don't think you will be needing. So what is happening in PCR? Now, polymerase chain reaction is a procedure where 3 steps are taking place. First, the breaking of the DNA double strands into single strands, then the duplication of these markers or creating multiple copies of the target markers that are radio labeled and that fluoresce under any incident light energy, and then annealing of these chains to form multiple copies of DNA. So PCR chain reaction is a technique to make many copies of a specific DNA region in the organism or in the test tube only. You don't need to take the DNA out of the test tube to create it. When you're creating something in the place where it is present, it is known as in vitro. Okay, so PCR relies on thermostable DNA polymerase, Taq polymerase, and requires DNA primers designed specifically for the DNA region of interest. So in CODIS, since we are studying 26 markers, we know the sequences of these 26 markers, right? We know the nucleotide sequences, the tandem repeats that are there in the beginning and the end of these markers. We can easily create templates of these markers that are radio labeled, and we can add it to the PCR liquid mixture so that they go and attach to the recognition sites of the markers that is present in the forensic sample. And then we can create multiple copies of these radio labeled markers and ultimately analyze it in the genetic analyzer to understand the polymorphic state of these markers. Ultimately, we can identify the marker position. We just need the starting point and the end point of these markers that are tandemly repeating, and we can create that radio labeled templates which can go and adhere to the target site and create multiple copies of such DNA markers in the PCR. So the PCR can be used as a tool in genetic fingerprinting. This technology can identify any one person from millions of them. Am I clear? Is this clear?
Yes, ma'am. Yeah, ma'am.
Right. So too much of information for today. I hope you guys might be having a lot of questions. Kindly ask me if you have any doubts or questions. Any doubts, any questions on this topic? Because 45 minutes of excessive data has gone, and I guess you all are undergraduates, right? Oh, MSc student here. So I don't know how many practical sessions of DNA you have had in your college. So let me know if you have any doubt, if you have any queries, if you want to ask me anything. We still have 20 minutes, 15 minutes left. So we can have that discussion if you want. Are there more people today? I guess there are more people today. I see 21. When I say I have discussions, people start leaving meeting. No doubts? You can unmute yourself and ask. Don't type. Unmute and ask. No doubts? Nobody is saying anything. Ganesh and Vagdevi, you guys were very active the other day. What happened today? No questions?
Actually, ma'am, we had biology in our previous semester. Like in April only, we wrote exams.
Sorry, what?
In April only, we wrote exams, ma'am. Forensic biology.
Okay. So
It's more like
you clear with all the doubts?
Yes, ma'am.
Okay. So how about you ask me about career guidance or anything else? Because I'm done with my topic here, so I don't want to go. I hope I did not go beyond what was there in your syllabus. I hope today's topic was not something that went up above your heads. I hope you all were able to understand.
Yes, ma'am.
So anything else that you want to know that we could not speak about the day before? Yes, Ganesh, I see you unmuting yourself. You have a query?
Yeah, ma'am. Ma'am, like these are the things that are currently going in India, or like it's common for everywhere. Like I want to know.
This
Like,
DNA examination. So DNA examination,
same
basic principle.
examination is going in every or like,
Everywhere around the world, either plus or minus, they're doing the same thing more or less. In international investigation and in Indian investigation, there's something known as the rapid DNA technology that has come up. Rapid DNA technology, I'm writing it in the chat box. So rapid DNA technology is basically used for crime scene samples. So instead of sending these DNA samples to the FSL lab, in 90 minutes, they create a profile of samples that can be degraded in transportation. So they take 90 minutes, they take sample at the scene of crime, and during the entire investigation process, while you are investigating the scene of crime, they keep these the rapid DNA machines on, and they create profiles of crime scene samples, and then they send these profiles only to the FSL profiles to the FSL for just analyzing these profiles. They just analyze these profiles. What FSL is doing is they are just analyzing these profiles that have been created at the scene of crime, so that the entire extraction and cleaning and amplification, this whole procedure, the whole procedure that takes up to 4 to 5 days, is reduced to just 90 minutes at the scene of crime. And the only scientific work of the FSL that remains is analyzing the genetic profile that has been sent by the person at the scene of crime and match it with the reference samples of the individual victim or accused, and just give an opinion as to whether these two profiles match or not, and whether that profile belongs to the individual accused or not. So this is the only thing that is coming up extra right.
okay, ma'am. Ma'am, can you help me to know? Can you help me to detail about this trace DNA?
Trace DNA is or touch DNA or any trace element that is present in around you. There are DNA components that are suspended in the air when you are sneezing, when you are sweating. There are aerosol forms of DNA, and this touch DNA can be collected and subjected to rapid DNA machine, and partial profiles can be created based on the basis of such minute quantity of DNA that is there. So basically, trace DNA is not something different. It is the DNA only. The principle remains the same. The separation principle, the genetic marker principles, the CODIS principle, the profile principles, everything remains the same. It's just that the quantity of DNA reduces, and we have advanced machinery that is able to detect such less amount of DNA. Basically, in laboratory, at least the minimum amount of DNA that should be there for autosomal DNA is somewhere between 800 picogram to almost 1 nanogram of DNA per microliter. So that quantity is needed. But in case of trace DNA, even 50, 60, 100 picograms of DNA can be analyzed. So that is the only difference.
Okay, ma'am. Thank you, ma'am.
Anybody else? No? Okay then, if you guys have no other queries, we'll end the session for the day. I am basically done with all the forensic biology aspect, and if you guys have any query, you can connect with me through email and number. I've shared my number also the other day, and we'll see you later. Okay, have a nice day. Take care.
Madam,
Yes.
ma'am, is there any new advancements came in this DNA sequencing or DNA
Advancement
fingerprinting
what?
techniques? In DNA fingerprinting techniques, like, is there any new advancements
I
are
told
coming up?
you NGS and mitochondrial DNA, NGS next generation sequencing and mitochondrial DNA for maternal DNA. These two are the only advanced. NGS, there is nothing beyond NGS because NGS is working on the Human Genome Project principle. They will do nucleotide by nucleotide profiling of your DNA and the entire DNA can be profiled. What do you want more than your entire DNA being profiled?
Okay, but ma'am, actually, in previous slides, I saw something like disaster victim identification, something like.
That is just the application of DNA profiling. The instrumentation is not changing. Instrumentation is the same. DNA has its application everywhere. Disaster management, victim identification, in case of mass disasters, mass deaths, train accidents, airplane accidents, cold cases, anthropological cases, even in cases of burial also we have 1000, we have people scientists working on 10,000-year-old genes right now, and they have recreated a species which was extinct 10,000 years ago, right? So DNA has its application everywhere. Disaster management system is just an application of the traditional principles of DNA profile.
Ma'am, in such cases of disaster victim identification, like a mass disaster is happening, how can they
Yeah,
identify those people?
we just create the profile and put it in the index system. See, in India, we do not have a DNA profile. We do not have a centralized DNA system. But in America, they do have every individual, every citizen of that country has its DNA registered. So if there is a disaster happening, and they're getting body parts like tissue, bones, and they're creating DNA profiles from it, and they're entering it in the database is immediately going to point out that who the dead person is. So they're just ultimately identifying from the help of DNA profile.
And then why our like people are not
Because
working
we
on
have
getting
right to
the database?
privacy. India has the right to privacy wala clause in their constitution. So the DNA bill is trying to be passed in the assembly, but it is not happening because it is breach of personal safety. That's why. So Indian laws do not allow it. We cannot share our Aadhaar data. The government is not allowed to share the Aadhaar data. How do you think they are going to allow the sharing of DNA profiles? No individual is. You cannot even force an individual to undergo a DNA examination in case of any forensic investigation also. An accused and victim should give a consent before undergoing any DNA examination. If the person is not consenting, you cannot force it. Unless and until you get a judicial order which compels them to undergo a DNA examination, a person can outrightly deny its DNA examination. So India is different from America. The
Yes,
policies and
ma'am.
laws are different.
Yes, but with some certain protocols and with some restrictions that
That is going to
only.
take time. It is going to take time. Creating a DNA database is going to take time. Maybe another 10 years.
That's only my question, ma'am. Like,
So
for
that is
more
at the policy
accuracy
making
in these
level, na?
investigations, we need that, na, ma'am.
Sorry, what? What?
For more
Sorry?
accuracy
Your voice.
and for speedy results, we need such kind of
Yeah, but that
databases,
is
right,
all
ma'am?
about the policy makers, no? The politicians and the policy makers, they need to make such policies. We can't do anything about it. Can we? We can't. We can just put our representations to them. Decision making and law making is their forte. not ours. Our job is to work on the guidelines that are provided to us. We cannot work on the policy making or the law making. That is on somebody else's shoulder, right?
Yes, ma'am.
Anybody else has any other query? No other queries? Okay then. See you guys. Have a great day. Good night.
Good night, ma'am.